afternoon, everyone. My name is Chloé, and I'm currently working at École des Mines, Center for Engineering and Health. And I'm going to present you today the project we do with the University of Saint-Etienne about the characterization of the mechanical interaction between smooth muscle cells and their collagen-based hydrogel matrix using in vitro traction for microscopy. So, yes, my main activity focuses on the aorta and the ascending aorta. The aorta is composed basically of three layers: the external layer, the internal, and the media, which represent 2/3 of the total wall thickness. The media is mainly composed of smooth muscle cells and elastic fibers. Smooth muscle cells are the main cells responsible for the active and passive response to external mechanical forces applied on the aorta itself. Contraction of smooth muscle cells is created via the actin myosin filaments, which slide against each other to create contraction. So, aneurysm is one of the most deathful cardiovascular disease. It consists of an enlargement of the aortic diameter of 1.5 times compared to a healthy diameter. And so far, this is how surgeons identify an aneurysm. The problem of aneurysm is there is no more precise understanding of how it's created and how to detect it in advance. So basically, most often we have a rupture of the aorta, which happens, but has been undetected because the size was not large enough to run examine. So the idea is one of the phenomena causing aneurysm is the disturbance of the phenotype of smooth muscle cells. As I said before, the main layer of the aortic wall is composed of smooth muscle cells, and what we observe is in case of aneurysm, we're gonna have different phenotypes for these cells. What happens because of biomechanical changes or genetical condition, the phenotype of smooth muscle cells is gonna be changed into synthetic phenotype. Synthetic cells is gonna stop contracting and instead is gonna remodel the extracellular matrix around the cells. So this leads to an imbalance of the composition of the tissue in the aorta, and in particular, we observe an increase of collagen content, an increase of the matrix degradation, and a loss of the contractibility of the tissue. This way, the wall is becoming weak, and its mechanical property changes, which leads to a higher risk of rupture. So, few things have been studied in terms of smooth muscle cell behavior and their change in biomechanical properties. And one way to assess the contractile behavior of the cells is traction for microscopy. It consists of taking two pictures of the cells in their environment. So you take one picture of the cells in the initial state and one later when the cells are contracted, for instance, and you're gonna compare the a special pattern that will be in the matrix, and you will be able to derive the deformation of the matrix and then to derive the traction forces that the cells apply on the matrix itself. So the study on smooth muscle cells traction forces has been pretty recent, and mainly it's been done in 2 dimensions. So here you can see the result for an healthy cells and an aneurysmal cells, and we observe that the cells do not apply the same amount of force on their matrix, which can indicate that it could lead to further signal to other cells and trigger and enhance the process of degeneration of the tissue. So as I said, so far, most of the studies on traction forces have been applied in 2D, but this is not a good representation of the in situ cells. So the idea of my project is gonna be to study cells in 3D, and in particular to have and to develop an imaging of these cells in 3D, and then using machine learning, be able to characterize the matrix and cells interaction mechanics, and to help like the prevention and the treatment of this cardiovascular disease. So the project gathered different work packages, which are quite different, and I'm going to review all of them, and I will present you the result we have so far with this project. So the first package was, first of all, we needed to develop a 3D model for the investigation of the traction forces of the cells. So this has been done using collagen-based hydrogels. We wanted something very simple to start. Then a second package is about micro-patterning the collagen-based hydrogels, because you can apply traction for microscopy using an external pattern like fluorescent beds, for instance. But the idea would be to be able to mark the hydrogel itself with the laser, so we don't integrate into the matrix any external element which is not existing in the real life. The third axis of the project is then to perform traction for microscopy with confocal and or a light sheet in order to have like the average mechanical force applied by the cells. Then we would like to go more in detail, both in spatial scale and temporal scale. And for that, we want to combine the previous result with inline holography, which would allow us to go to sub-seconds and have a very low, smaller resolution. And finally, all the results, experimental result, would be put into an automatic learning of a mechanical model for the cell-matrix interaction, which would allow us then to be able to apply this mechanical model on patient cells to identify whether or not the cells are healthy or present a risk of aneurysm. So this is like a broad project, and so far I'm gonna present you to start the first one. So basically, we're starting from scratch. The previous work, as I said, was like traction for microscopy, but in 2D. So the problem of cells is like a cell is gonna adapt its behavior based on its environment. So it's both dependent of mechanical property of the matrix, but also the microstructure. So we needed to have like a 3D model which would maximize the contractile morphotype and phenotype of the cells in order to traction force microscopy. So the first step was to design a hydrogel which would be optimal for the study we want to proceed. Then we wanted to understand the impact of the different parameters of the matrix on the smooth muscle cell culture and the population. And finally, like we wanted to understand how the mechanical property of the hydrogel also would impact the smooth muscle cell culture. So basically what we did is to use basic collagen hydrogel because wanting something simple to start. So we created a different dilution of collagen, and we add some control, so acellular gel that we created. Then by using cell culture, we could seed the hydrogel. So our population was homogeneously scattered into the matrix. And then we were able to, we waited 5 days for cell differentiation. And we studied the hydrogen in different ways. So for the acellular gels, we did rheological tests. We did some staining of the collagen fibers as well, in order to have an idea about the microsculpture shape. And finally, we stained the cells to see their morphotype. We varied, designing the hydrogel, we varied the pH with 3 different values in between the range, in vivo range. And we tried collagen dilution of 2.5 to 10 milligram per milliliter. And we also seeded the hydrogel with 3 different density to see the impact of the initial cell population on the behavior of the overall population. So this resulted in 60 tests. And as I said, we used basically, in order to limit the variation in the dilution, we used 3 different repetitions coming from the same batch. And adding then benzyl media for the acellular gel, and we use cells at pH 10 for the hydrogel, with the seeded hydrogel. Mechanical test was performed at 3D Fab Lab in Lyon, and we tested the hydrogel in shear and compression in 2 decade range. The first biological result, the first thing with the biological results, like we wanted to basically have an idea of how was share the population and which phenotype of morphotype we were able to obtain, depending on the different property of the hydrogel the cells were seeded in. So for that, we defined 5 different morphotypes based on the length and the shape of the cells. So we had the long cells, over 100 micrometers, the short one, and the round that we associated to apostosis. So we were able to record for each composition of hydrogel the repartition of the different morphotype within the population of cells. And then we observed and compared the proportion of each phenotype compared to the properties of our hydrogels. So what we observe is, to start, is like we had like the whole quantity of cell decreased above 5 milligram by millimeters collagen concentration, meaning that when the hydrogel is too dense, cells population tends to not to die basically. Similarly, we observed that the cells tend to elongate for low collagen concentration and for high cellular density. We observed in reversely that the dead cells, so the cells E, the round one, that they tend to increase with increase of collagen concentration. And finally, the length of cells seems to be inversely proportional to the collagen concentration, meaning that more fibers we have, the shortest the cells were getting. We measure the mechanical properties of the hydrogels, and we find like consistent results with the literature, so proportional increase in modulus and viscosity with the collagen concentration only. And looking at the microstructure, that's where we were very surprised, because not only we observe the expected change of fiber length and pore size according to the concentration, but we observe also 2 different patterns that you can see on the right. So this one was more at the bottom of the hydrogel, and this one on the top. And what we wanted to ensure with these images was like our hydrogel was isotropic. The thing is, we observed that we had like a limit effect at the bottom where the gel was attached, but also that despite the fact that within the hydrogel we had isotropy, at the surface we observed that an alignment of fibers in some cases. So this is probably due to the manipulation of the hydrogel, where the medium is gonna just move the surface fiber and align them during like manipulation. So the conclusion of the experiment allowed us basically to have the best configuration for having contracted cells. So the idea was like, if I have a quantity of cells, I want them to be able to undergo traction for microscopy. So I want them to be contractile. I want them to be at the maximum in quantity as I can have. and at the minimal loss as possible. So we discovered that for having such a gel, so a gel which would maximize contractile behavior, would be like with the maximum cell density as possible, and for the lower collagen dilution. That was the two conditions that would maximize our contractile population. Also, the pH was not significant. And so basically, this was the first work on smooth muscle cells in 3 dimensions, and we had like some limitation. We used like advanced passage for cells, so we were at P11, but we checked they were still contractile, so this did not modify our result. Also, the measurement of the length was more approximative than in 2 dimensions because, of course, the cells were not aligned horizontally most of the time. So we just measure from the picture. We did not like do some projection and some computation. Also, imaging the collagen fibers was quite challenging, specifically in deep, because the fluorochrome, it was difficult basically to obtain an homogeneous enlightening of the hydrogel. So this is something we're going to have to work on. And also for the mechanical test, having a homogeneity of the surface was a challenge, but we managed to overcome it. So to conclude, this first part of the project allow us to have an ideal matrix for further testing of smooth muscle cells. With this hydrogel, we're going to maximize the quantity of contractile cells. We observe that the cell death increase with concentrated, with the concentration of collagen. And basically, we also observe that the cell length was limited by collagen concentration. Although we don't know if it's of the mechanical property of this gel or if it's about the pore size, for instance. So the second part of the project is going to be to create micro patterns within the hydrogel. So for that, we're going to collaborate with Manutech-SLEIGHT, which will like have a femtosecond laser. And we're going to go and apply different technique on the hydrogel in order to create marker, but based on the architecture of the matrix, so using the collagen fibers. The different option was going to be basically adding densification of the fibers locally, but also adding removing collagen locally as well. And the third part that we've started to work on is the traction for microscopy for smooth muscle cells. So the idea is to first develop DVC, so digital volume correlation algorithm, and optimize them for the cells and for the hydrogels. So one of the axes of this project is going to be the comparison of the traction for microscopy using beds, fluorescent beds, and using the fibers of collagen as a pattern. And this will give us the average force supplied by the cells on their environment before going into a more precise analysis with inline holography. So that is everything for me. I would like to thank my co-author, and if you have any question, I'm available.